Hit or Miss, Influenza Vaccination by Gokul Gowri. So before I start my presentation, I'd like you to see a picture of a flu virus. This is a flu virus. The pin-like objects on the outside are the surface proteins. The red ones that look similar to mushrooms is hemagglutinin, and the blue ones are neuraminidase. These little donut-like objects around, those are also surface proteins. That's called the M2 protein. It makes up the ion channels. M2 matches about 85 to 95% in all flu. It also mutates very slowly. This green on the inside is the RNA, the genetic material. Question. So in this project, I had two questions. The first question was, how does the degree of the match of the amino acids in the surface proteins in the virus and the vaccine correlate with the severity of the flu in the population? So what this means is, how does the similarity of the virus and the vaccine affect the severity of the flu? My next question was, will the universal vaccine reduce severity? And what this means is, how would a new proposal for a vaccine affect the severity? Introduction. So you may think that the flu is not very serious. You just get sick for a weekend and then you're better again. But it really is a really severe thing. Every year in the United States, 30,000 people die from the flu. The reason we get a new flu shot every year is because viruses tend to mutate quickly, which means the virus doesn't stay the same from year to year. The vaccines we get are trivalent, which means they protect against 3 different strains of the virus. So the vaccines are made a year before because it takes 8 to 10 months to make the vaccine, so they need to predict long before the season actually starts. So the scientists don't always get exactly what the flu is going to be. So sometimes the predictions aren't accurate and the vaccines don't work as well. So there's something called an antigenic shift. An antigenic shift is when a virus mutates so much that it changes strains completely in 1 year. Antigenic shift can cause scientists' predictions to go wrong because scientists have predicted probably what the same virus was, not a new one. And this can cause a pandemic. Our 2009-2010 swine flu pandemic was caused by an antigenic shift. To solve this, there has been a proposal for something called a universal vaccine based on the M2 protein. Since the M2 doesn't mutate very quickly and also matches very well between viruses, people have proposed that making a vaccine with this would protect against antigenic shift pandemics. So I talk about the similarity of proteins a lot in my project, and I determined the similarity of proteins by something called BLAST, which is Basic Local Alignment Search Tool, which tells me the percentage of matching amino acids between 2 proteins. Procedure. So, first, I found the most common strains of the virus and the vaccine for the years 2001 to 2013. I got this from the CDC, Center for Disease Control website. Next, I went to the National Center for Biotechnology Information website, and I got the amino acid sequences for hemagglutinin and neuraminidase. I blasted these to find the similarity of the proteins. Next, I went to the CDC website and I collected the data for severity. For flu, there's no specific unit for severity. The CDC determines severity based on a combination of 5 statistics, which are pediatric mortality, hospitalization, percentage of death attributed to flu, percentage of outpatient visits attributed to flu, and percentage of positive lab tests. Using these, I created 5 scatter plots. I did regression analysis and took the R-squared to find the correlation between the percentage of matching amino acids and the severity. Next, I went back to the NCBI website and I got the amino acid sequences for M2 and I blasted these. I took my percentage of matching amino acids and I put it into my regression equation to estimate the severity. Results. So here are my estimated severity had we used an M2 vaccine. In the parentheses are what the real statistics were. So in most years in all years the severity was decreased, and in most years it was decreased greatly. In 2009-2010, our swine flu year, the original hospitalizations was 42,000, and my estimated hospitalizations if we had used an M2 is just under 18,000. That's almost 25,000 hospitalizations saved. That's about 60%. A few exceptions to this is this one right here, in which the percentage of positive lab tests was 19% in real life, and my estimate was 22%. There might have been other factors that could have caused this percentage to be really low, so there are obviously some exceptions. So these are my scatter plots. In my hospitalizations regression, I got a 0.9517 R-squared value, which is very high. That means there's a very strong correlation between the percentage of matching amino acids and the hospitalizations. In outpatient visits, I got a 0.633 R-squared value, which is a fairly significant correlation. For pediatric mortality, I got a 0.757 R-squared value, which is also significant. In positive lab tests, I got an R-squared value of 0.7073, which is also a significant R-squared value. That means there's a good correlation. But in mortality rates, I got a 0.0731 R-squared value, which means it wasn't very significant. So this is because the mortality rate statistic not only had the people who died from the flu, but also the people who died from pneumonia. Severe flu can lead to pneumonia, but there's still independent pneumonia, which can cause this statistic to be irrelevant to my investigation. So even though only 4 out of the 5 criteria were significantly correlating, I still concluded there was a significant correlation between the percentage of matching amino acids and the severity. Discussion. So from my results, I determined that as the percentage of matching amino acids increased, the severity decreased. I also determined that had we used the M2 protein vaccine, the severity would have been lowered. I determined that the M2 vaccine would be more effective than our current hemagglutinin and neuraminidase vaccines because they have a much higher percentage match of amino acids. M2 can prevent pandemics because a lot of pandemics are caused by antigenic shift. And antigenic shift is caused by the mutation of the protein you're using for the vaccine. So, if we used an M2, we would have less antigenic shift pandemics because the M2 protein doesn't mutate quickly. In various lab tests and animal studies, they've determined that M2 protein vaccines have immunogenicity, which means they provoke a reaction from the host. One of the challenges with the M2 vaccine is that it's very shallow on the membrane of the virus, so it can be difficult to recognize for the body. But people have determined that if you increase the concentration of M2 in the vaccine, this problem can be solved. So, since the early ages, vaccination hasn't advanced much. The first form of vaccines came 3,000 years ago by the Chinese, who took the pus from a cow which had a form of smallpox, and they let citizens inhale it. Since then, our concepts for vaccination haven't advanced much. We're using the same concepts, just on a little bit more of a cellular level. I think that we can improve this and maybe even create a new way for vaccination, which would greatly improve vaccines. Many people say this is difficult or even impossible, but if you look back 100 years ago, people also said it was difficult or impossible to land on the moon. I think that because many people worked at landing on the moon, we were able to land on the moon. So if many people worked on creating a new vaccine, we could also create a new vaccine and take our first step towards eradicating infectious diseases. So right now, we need more awareness. Because even if we have an effective vaccine, we need people to take the vaccine. A vaccine is useless if no one gets it. Today, many people forego getting the vaccine because people think it's linked to autism and other congenital malformations. But these have been scientifically proved incorrect. So I think if we can get that out there and make more people learn about it, more people will get the vaccine, and we can take our first step towards eradicating the flu.